So I think we'll get started. Hello. So it's a pleasure to welcome our guest today, Dr. Sora Shin from the University of California at San Diego. Dr. Shin is in the neurobiology section in the Division of Biology at UCSD. She did her doctoral work in medical science at Yonsei University College of Medicine in Seoul. And then she came to the U.S. to do a postdoc at UCSD, where she currently is in the lab of Bin-Kook Lim, and has had a very productive run there. She recently won a Kavli Institute for Brain and Mind Innovation Grant, which are very competitive. And she's also, along the way, in addition to her research and publications, has developed some intellectual property, filing patents, for example, developing a new method to test social dysfunction in autism, looking at the Drd3 dopamine receptor signaling pathway. And she's had a very nice run of important papers over the last couple of years, including a publication in Neuron, a publication in Cell, a publication in Nature Neuroscience, and one in Metabolism. For her doctoral work, she studied metabotropic glutamate receptors, particularly the mGlu-R5 signaling pathway in the nucleus accumbens, and its role in development of depression and stress resilience pathway in this nucleus, and provided some really, I think, interesting new insights on circuitry and signaling in this disorder. For her postdoctoral work, she's been doing a variety of interesting things, including generating multiple viral tools to analyze circuit dynamics in the lateral septum. And she has been looking at the role of the lateral septum in early life stress-induced social dysfunction. And she's also been able to utilize and apply in vivo larger scale monitoring of neural activity in freely moving animals. And this work has been focusing on what I think is her main and consistent interest, and that is she's interested in stress-induced pathophysiological mechanisms that can involve signaling pathways and behavioral dysfunction, and particularly interested, I think, in neuromodulatory pathways, including in the lateral hypothalamus, where she's looked at early life trauma-induced effects leading to things like binge eating and ultimately obesity. So it's really a wonderful series of converging technologies and approaches that brings together leading-edge neurobiological approaches to understanding complex behaviors. So we're really happy to have you here today, Dr. Shin. Welcome.
Is that work? Can you hear me? Okay. Thank you for nice introductions. So today I'm going to talk about stress, which is a really common environmental factors. But as you know, the toxic stress and trauma can damage our mental and physical health, but still we don't know how it happened. So during my Ph.D., I studied stress experience after becoming adult, and also its resilience mechanisms. But when I started my postdoc training, I became interested in some different type of stress which has such a strong impact throughout the life. That is early life stress. So every adult has their own history of childhood experience. So no matter it was good or bad, the childhood memory is almost permanent. Particularly once we're exposed to stress or trauma, the wounded emotions has been stored as this crying in a child, so that affect our feeling, thought, and behavior outcomes even after we become adult. So then what types of early life stress we can think about then? There are some categorized adverse childhood experience, or ACE, including emotional and physical abuse and neglect, and also the life-threatening trauma and household dysfunctions with mental illness, substance abuse, or parental divorce, and the witness of domestic violence. So from 1995 to 97, there was a big survey asking adult people about their history of exposure to adverse childhood experience. So it was total 10 questions. So for every yes, you get point on your AC score. And they found that the people who have at least one AC is 60–70% of total populations, meaning that this early life stress is extremely common and prevailed in our society, unfortunately. And also they found that there was strong correlation between early life stress and health outcomes. The higher AC score, the worse health outcomes. For example, the people who have AC score 4 or more, they had much higher risk of health disease like depressions and addictive behaviors, and also high suicide attempts, and also the physical health, including lung cancer, heart disease, and obesity, and also even early death. So this study is obviously showing us that the early life stress is a critical risk factor threatening our life and our health in later life. But the question is that still we don't know how the different type of early life stress can drive to its pathological symptoms. We always know there is strong correlations, but how it happened? What is the underlying neural mechanisms? And that's the question I had in this study. So firstly, I'm going to look at the stress of neglect, particularly the emotional neglect. So it's just a few years ago, in England, a new law, it's called the Cinderella Law, was in effect to stop parental emotional neglect. So based on this law, not only the physical abuse or the sexual abuse, but also the parents who didn't show enough love and kindness to their children, like the wicked stepmother of Cinderella, then they can go to jail for up to 10 years. So why we are getting seriously considering about this issue? I think the part of the reason is that we become aware of that exposure to emotional neglect in childhood can bring tremendous change in our life. For example, it can induce long-lasting change in gene expressions of neuromodulator or its receptors by changing the epigenetic modification or neuroendocrine system. And also in brain level, it can change the structural difference with different size or different neuronal activity. And behaviorally, it can induce a number of psychiatric symptoms, and among them, the social dysfunction, which is a core symptom of depression, was suggested as a main problem because most of those children have trouble in emotional regulations, so they have problem in interpersonal relationship. And I think we can find a representative example of this from the historical event. So from the 80 to 90, there was a Romanian dictator who wanted to make population growth. So he banned all the abortions, contraceptions, and he let the parents who have less number of parents pay the huge tax. So because of that, at the time, the birth rate is quickly going up. But at the same time, a number of babies were abandoned and institutionalized because most of the parents couldn't take care of all the baby due to the economic difficulty. So at the time in institutions, more than 20 babies were assigned to single caregivers. So those babies were exposed to early psychosocial deprivation with lack of caregiver stimulations and emotional investment. And later, these babies were adopted to better place, but it turns out that a number of those babies showed social difficulty. They didn't respond to social stimulus, and they have poor communications, and also they showed repetitive behavior, which are the autism-like features. And also, they have smaller brains than normal children with enlarged ventricles and shrunken cortex. We can see how destructive this stress is. But still, there are a number of questions we need to address. Still, we don't know which specific neural substrate is critical for this process. And also, what's the molecular function or the neural activity has been changed in their brain. And lastly, we wanted to ask whether we can think about any possibility of recovery. And these are questions I got to have when I started my postdoc training. So in order to study this, firstly, we need to have the animal model system that mimic this type of situations. So we apply early social deprivation stress, meaning the separating a mouse pup from both their mom and little mate for 3 hours per day during the first 2 weeks of their life. So during that time, these mouse pups get exposed to complete social isolation without mom's care or any physical touch. So we call it as early social deprivation, or ESD. And when they get to full adult stage, we test their social behaviors. So we test 2 aspects of social behavior. The number 1 is sociability, which is the preference for interacting with other conspecific animals. And we can test it using this 3-chamber arena with 2 sides. One is just empty, and the other side containing stranger mouse inside of this mesh case. So we put the testing animal in the middle arena, and then we can measure their social preference level by looking at where they prefer to stay more. And the second test is reciprocal interaction test. So in this test, we used freely moving stranger mouse, so we can detect more direct social interaction behavior, like nose-to-nose contact, body sniffing, and following behaviors. So firstly, in the 3-chamber test, we found that the control mouse, they prefer to stay in this mouse chamber, and they are interacting with their friends because they have natural social motivation. However, ESD mouse, they tend to spend more time in these empty chambers rather than interacting with their friend. So when we calculate their social preference level based on the resident time, we can see the ESD mouse had much lower social preference level compared to control mouse. And also in the reciprocal interaction test, ESD mouse consistently showed lower social interaction time. Particularly, they spend much less time in the following behaviors, meaning that they prefer to stay away from the other animals. So based on this data, it seems like after exposure to early social deprivations, they get to have reduced sociability. And the second test we try is the social communications. So generally, rodents has their motivation to communicate with other animals. For example, when male mouse encounter female mouse, they produce ultrasonic vocalization call to make the female be interested in him and to facilitate their mating behaviors. So we can measure their communication level by counting how many times they produce call and how quickly they make a first call. So interestingly, ESD mouse produce much less number of calls, and they took a long time to make a first call when they met a female mouse. Even though they have normal sexual behaviors, but only control mouse produce active courtship song before their mating, but ESD mouse didn't try to communicate, and they just tried to mating behaviors. It seems like they also have severe defect in social communication part. So then our question is that which specific brain area is involved in this behavior phenotype?
Yeah, that's a quick question. So you showed us the data on the following, and now on the vocalizations. Are those linked? In other words, do you have data to suggest that their lack of participation, for example, in the following behavior is due to some disruption in their communication?
I think it depends for the following part. It's an interaction between male-to-male interactions, and they show the reduced following. But for the female, they didn't show some less following behavior. they showed the following amounts, but communication was disrupted. So we apply staining CFOS, which is neuroactivation markers. So particularly in order to look at specific brain area most responsible for social behaviors, we sacrifice a mouse after their social inertial behaviors, and then we quantify CFOS from the several brain area which are known to be involved in social behaviors. And from this experiment, we found very interesting brain area that is lateral septums. So this area showed most significant increase of CFOS after they had social interaction behaviors. But more interestingly, ESD mouse didn't show any response, even in the same brain area. So it seems like the lateral septum is important brain area mediating the social dysfunction behavior of ESD mouse. And previously, the lateral septum is thought to be important for modulation of motivated behaviors and social preference. And also it contains a number of cell type of neuron expressing neuromodulator and its receptors. So then next, we wanted to ask which specific cell type of neuron inside of lateral septum is important for mediating these behaviors. So we assess mRNA level, particularly the different subtype of receptors for dopamine, serotonin, oxytocin, glutamate, and glucagons, which are abundantly expressed in the lateral septum. And we found that ESD mouse have much reduced expression of dopamine receptor 3 in the lateral septums. Interestingly, the Drd3 molecule is heavily implicated in many of human psychiatric diseases, including autism.
I'm curious, the oxytocin receptor, no difference at all?
No difference at all. Yeah, I was surprised too. Yeah, because in previous study, the oxytocin receptor in the lateral septum has been studied and their social preference, and it affects their behavior. But in my study, in terms of expression level, it doesn't change. Yeah. Yeah, Drd3 is heavily implicated in psychiatric disease, including autism spectrum disorder in human clinical study. So we presume that Drd3 lateral septum neurons may be the important component to mediate the social dysfunction of ESD mouse. So then next, we wanted to ask whether this specific neural population is relevant for social behaviors. So we decide to monitor endogenous activity of Drd3 lateral septum neurons by expression of GCAM6, which is calcium indicators. So when it binds to calcium, it can produce EGFP fluorescence, so you can monitor it. And particularly, we clone this GCAM6 between two different kinds of LOXP site that is called DIO strategy, in order to make the selective expression of GCAM6 in the presence of a created combinase. So for example, if we inject AAV virus expressing DIO GCAM6 into the lateral septum of Drd3 cre mouse, then it will allow expression of GCAM6 only in the Drd3 positive neuron in the lateral septum. And then we implant a small size of miniscope into the same area to monitor intracellular calcium dynamics. So this is the sample video we're imaging from the Drd3 lateral septum neurons. So we can detect the dynamic calcium transient from individual neurons. So one of the strengths of this method is real-time monitoring of neural activity in the freely moving animal. So it can make it possible to correlate between the neural activity and a certain type of behaviors. So in this case, we are going to look at their social behavior. So we compare two different situations. The one is the social context, because we add social stimulus. But the other case, we add fake mouse, which is not the real mouse, it's just mouse toy. The mouse can approach and make a physical contact, but that's not the real social behaviors. And then we monitor calcium activity before and after we add each stimulus. So from the analysis, we can get this type of raster plot. So each row means individual neurons, and each one tick means single calcium transient. So we can see increased number of calcium transient after they exposed to social stimulus. Particularly when they make a social bout with this transient mouse, we can detect the calcium transient from Drd3 LS neurons. However, in the presence of fake mouse, there's nothing changed. Even when mouse approach and make a physical contact with fake mouse, these neurons didn't respond to that behaviors.
So, sorry, question. So how do you know that whatever signal you're getting there is not related to something as simply as an olfactory cue, for example? I mean, putting in the context of a social percept, which of course can have many components to it, sensory components. Is there a way to rule that out, or do you know that the LS won't respond if you just put in a smell of the mouse, for example?
We haven't tried the sensory part because further of our focus is comparing control and ESD mouse. But when we test their basal behavior, especially also we also checked the olfactory part, but we couldn't see the difference at that part. So we didn't check out that how its sensory part affect the social behavior. Yeah, but it's an interesting point. We can try.
I mean, is it known from previous either neurophysiological recordings, optical recordings, whatever, in this area in lateral septum, that there are sensory olfactory responses or not, for example?
As far as I know, yeah, I couldn't report that part. Yeah.
Okay. Quick follow-up question. Are you including both kind of the following behavior as well as stopping and investigate? Like, are you separating out activity here in terms of?
You mean the? I also checked the following behavior.
So here specifically, are you excluding animals running, or are they stopping sniffing? Are you separating?
No, it's just freely moving, and yeah, they can contact. Yeah, that's it. So they didn't show any response for this fake mouse, so suggesting that Drd3 LS neuron activity is highly associated with actual social behavior rather than with general novelty or movement. So then how about ESD mouse? We already know ESD mouse have social dysfunctions. So then how do their neural activity would respond to this social stimulus? Surprisingly, there's no change. Even when they exposed to social stimulus, they are very calm. While control mouse consistently showed increased number of calcium transients, and they very sensitively respond to that social stimulus. So it seems like after they exposed to early social deprivations, the Drd3 receptor neurons get to have impaired neural activity, and presumably that may cause their social dysfunction in this mouse. So then we ask, what behavioral change would happen if we activate these neurons? So in this time, we express channel rhodopsin, which is the light-gated cation selective membrane channel. So if we deliver blue light on this channel, it can make neuronal activations by the depolarizations. And after this surgery, we perform two times social preference test. And the first one is just baseline behavior without light stimulations. But the other day, we perform the test again. But in this time, we deliver blue light for excitation. And this is what we saw. So when we deliver blue light, the ESD mouse expressing channel rhodopsin, they remarkably rescue their social preference level up to the normal range, suggesting that activation of Drd3 receptor neurons can rescue their social dysfunction of ESD mouse. So it seems like ESD, the early social deprivation, caused impaired neural activity in the Drd3 LS septum neurons, and seems like that is important component to induce the social dysfunction behaviors. But at the beginning, we also found that ESD mouse have downregulation of Drd3 signaling in the lateral septums. So then now we wanted to ask how these signaling chains affect to the behavioral change. Particularly, the Drd3 is a receptor molecule that can be modulated by pharmacological agonist or antagonist. So if we learn something about this receptor function in early life stress, then we can get great implication for the translational research. So for that, we decided to use Drd3 agonist, Ph.D compound, in order to upregulate this Drd3 signaling. And this is what happened. So after we inject Drd3 agonist with systemic injections, ESD mouse remarkably restore their social dysfunction to the almost normal range. However, this drug effect is based on the systemic injections, meaning that the drug will diffuse all the brain area, so it cannot tell us about the specific brain area information. So we try to disrupt Drd3 signaling only in the lateral septum by injection of shRNA, so that to see whether it can change this drug effect. Surprisingly, the rescue was gone, suggesting that the lateral septum Drd3 signaling is a critical component to mediate this rescue drug effect. So we confirm these connections, and then we have the last puzzle. We wanted to ask whether these two events are separate or connected to each other. How can Drd3 signaling change its neuron activity? So Drd3 receptor is GI-coupled receptor. So when it activated, it can inhibit PKA activity, and it can lead to the reduced phosphorylation level onto the GABA receptors. And it can facilitate the endocytosis of GABA receptors. So finally, it can produce reduced inhibitory synaptic transmission and also increase neural activity. This is the potential mechanism reported in the GABAergic neuron in nucleus accumbens shell. And we wanted to apply this idea onto the Drd3 lateral septum neurons because it's also the GABAergic neurons. Indeed, the activation of Drd3 signaling can reduce evoked inhibitory postsynaptic current. And this work was done by the Horia, the postdoc in our lab. And not only that, we can also find that the increased number of calcium transient after we inject the Drd3 agonist, meaning that activation of Drd3 signaling can lead to increased neural activity in the lateral septum. So we could complete this story. So this is a summary of the part 1. So we found that early social deprivation can produce the social dysfunction behaviors, and that is accompanied by impaired Drd3 lateral septum neuron activity, as well as downregulation of Drd3 signaling. And using the pharmacological activation and also oplogenetic activations, we can rescue their social dysfunction behaviors. And finally, we also found that the Drd3 signaling in the lateral septum, they can have the corresponding effect onto their neural activity. So in part 1, we saw the emotional neglect and how it leads to social dysfunction behavior, which is a core symptom of depression. And now, in part 2, we are going to see the trauma and how it affects our eating behaviors, and to produce the severe obesity. So the trauma is a severe shock or some life-threatening experience. And there are a number of human clinical studies suggesting that exposure to childhood trauma can lead to increased eating behavior with emotional hunger in later life. And actually, the large portion of binge eating disorder and obesity patients, they have history of early childhood trauma. So then we wanted to ask specifically what types of trauma is associated with these pathological symptoms. We can also find a clue from some tragic historical events. So this study showing a report some PTSD symptoms from the Kuwait children who were exposed to golfer during in 1990s. So particularly, they exposed to emotional trauma from death of family member and parental loss. And a long time later, it turns out that many of these children showed overweight and obesity symptoms based on their greater body mass index. And we can find a second clue from another tragic historical event. It's called Dutch Hunger Winners. So in 1944, near the end of World War II, when Nazi's army retreated from the Netherlands, they destroyed all the farms and bridges, docks to cut off all the way of food shipping. So people who living in there, they need to survive very limited food. And over time, the situation is getting worse and worse. So later, the English airports dropped the food in this area. So finally, this terrible famine was ended up. But during that time, there are a number of people who died, and also the survivors who were exposed to extreme hunger stress. And a long time later, surprisingly, people found that the survivor, particularly exposed to this famine when they were in mother's wombs or during the childhood, they showed high rates of obesity and overeating issue. Even 45 years after this event, this tragic early life stress was expressed as these pathological symptoms. So this is very surprising to me, and I really wanted to investigate the underlying neural mechanism of this process. But then the first thought came to my mind was that how can we replicate this emotional trauma and extreme hunger stress onto the mouse in the laboratory setting? So we tried to apply one-time strong trauma onto the mouse by separating them from mom and little mates. How long? It's 23 hours. So during that time, these mouse pups exposed to severe emotional neglect as well as physiological hunger stress. So when they get to, so at the time of weanings, they show the lower body weight than normal control. But over time, they compensated. So when they get to full adult stage, their body weight is pretty in normal range. So using this mouse model, we test whether they have binge eating disorder, binge eating behavior, or the obesity symptom, obesity-prone feature. So firstly, we want to look at the binge eating behaviors. So what is binge eating behaviors? This behavior is characterized by eating larger amount of food in a short period of time. And this is more like emotional eating, because even when they're not feeling physically hungry, they eat a lot without self-control. However, unlike bulimia, this binge eating behavior is not followed by any compensatory behavior, such as purging or exercise. So many times, this binge eating behavior is being associated with development of obesity. And the pathological binge eating behavior is characterized by the recurrent episode. And there is some previous study suggesting that the laboratory mouse can also have this binge-like eating behavior. So firstly, we wanted to test their protocol in our system. So we use high-fat diet containing high fat. So this food is tasty. So when we present both food at the same time, during 2.5 hours, mouse barely touched normal chow. They consume the high-fat diet. And then in order to induce binge eating behaviors, we remove high-fat food for 5 days and then re-expose them to the high-fat diet because this intermittent access schedule can boost animals' craving behaviors. Indeed, at the day of re-exposure, we found that mouse consume larger amount of food, which is almost double than the first time exposure. So even though they never exposed to some food restriction or calorie restrictions, but they consume larger amount of food in a short period of time, which is binge-like eating behaviors. And then our question is that how the mouse exposed to early life trauma would respond to this binge-like condition. So we apply both control and ELT mouse to this binge eating paradigm. And we found that at the first time high-fat food exposure, they both showed a similar level of binge eating behaviors. However, when we repeat this binge eating cycle by changing food from the normal chow to high-fat, on the second time, third, and fourth high-fat food exposure, ELT mouse shows higher binge eating behaviors. Particularly, they show the biggest difference at the last binge eating cycle, meaning that ELT mouse have stronger and sustained binge eating behavior than normal control. Not only that, when they exposed to chronic high-fat food exposures, ELT mouse showed accelerate weight gain change over the normal control. So their body weight is quickly going up over the normal control. However, when they exposed to chronic high-fat food, chronic normal chow, they both showed a similar pattern of weight gain change, suggesting that ELT mouse have high vulnerability to weight gain, especially when they exposed to high pallor food, not to the normal chow. But as we know, the weight gain change can also be affected by energy expenditure, not only by eating habit. So we test their oxygen consumption and the carbon dioxide production level while they exposed to high-fat diet. We can see the up and down change depends on the light and dark phase. The thing is that there's no significant change between control and ELT mouse, suggesting that the high weight gain change of ELT mouse is probably not based on their energy expenditure. So then our question is that how the mouse exposed to early life trauma showed the sustained binge eating behavior and high weight gain onto the high-fat diet, not to the normal chow. How do they have strong desire to eat this food? So firstly, we wanted to look at the specific brain area associated with this sustained binge eating behavior. So in this time, we quantify CFOS after each binge eating cycle. So among many other brain areas, we found that the lateral hypothalamus can be a great candidate to explain its potential mechanisms. Because in this area, after the first binge eating cycle, when both control and ELT mouse showed a similar binge eating level, they both showed a similar increase of CFOS. However, at the last binge eating cycle, when ELT mouse showed a stronger binge eating behavior than normal control, then we can see the CFOS response only in the ELT mouse brain. And the lateral hypothalamus is the area showing the most significant change of these patterns. So we presume that the lateral hypothalamus may be important the brain area to mediate the sustained binge eating behavior of ELT mouse. And previously, the lateral hypothalamus is well-known brain area, modulating feeding behavior and also stress response. And also this brain area contains heterogeneous cell type of neurons. So then next, we wanted to ask which specific cell type of neuron inside of a lateral hypothalamus is critical for mediating this early life trauma effect. So we assess over 20 candidate genes expressed in the lateral hypothalamus and also involved in feeding behavior and stress response. And among them, we found that ELT mouse showed reduced expression of leptin receptor in the lateral hypothalamus. The leptin is satiety hormone produced by adipose tissue, but its receptors are located in the brain. So its receptor signaling is important for mediate anorexic effect by suppression food intake. So we presume that maybe the leptin receptor lateral hypothalamic neuronal population is a critical component to mediate high binge eating and high weight gain phenotype of ELT mouse. So then we wanted to confirm this data, not only mRNA level, also in protein level, whether ELT mouse have impaired functionality in the leptin receptor. So in this time, we assess phospho-STAT3. So STAT3 is one of the downstream molecule of the leptin receptor. So when this receptor activated, it can phosphorylate the STAT3. So this is a good indicator to look at the functionality of leptin receptors. Surprisingly, we found that when we stimulate this leptin receptor by injection of leptins, the control mouse showed normally increased phospho-STAT3, but ELT mouse did not, suggesting that ELT mouse have impaired functionality in the leptin receptor in the lateral hypothalamus. So then we wanted to ask whether this molecular change is really matter to make their behavioral phenotype in ELT mouse. So in this time, without early life trauma, we just disrupt leptin receptor in the lateral hypothalamic area by using the expression of shRNA. And we found that the mouse expressed shRNA, they showed sustained and hyper binge eating behavior and also heightened sensitivity to weight gain when they exposed to high-fat diet, not to the normal chow. This is very similar with ELT mouse. So this data suggests that knockdown of a leptin receptor in the lateral hypothalamus can recapitulate the behavioral phenotype of ELT mouse. So it seems like after early life trauma, they showed downregulation of leptin receptor signaling in the lateral hypothalamus, and it seems like that is very important factors inducing high binge eating and high weight gain behaviors. So then how about the neuron activity? In previous CFOS data, we found that activation of a lateral septum in response to binge eating behavior. But in this time, we wanted to look at how the activity from this specific neural population is associated well with this behavioral phenotype. So in order to study that, we decided to monitor endogenous activity from leptin receptor lateral hypothalamic neurons by expression of GCAM6 into this area. And particularly, we monitor in vivo calcium activity every time the re-high-fat food exposure to see how this neural activity is correlated with binge eating behavior. And particularly on the test day, we present normal chow first, and 5 minutes later, we present re-high-fat food, and then we monitor the calcium activity before and after we add each food. So this is the sample video in the presence of normal chow. So they just approach and investigate the food, but they are not seem to be very interested in this food. And also at that time, the neural activity is pretty quiet.
Is that, are we actually seeing a time-lapse movie of the in vivo imaging up there?
Yeah, it's a
Just
time-lapse.
nothing's changing at
Not,
all?
yeah.
Okay.
However, in the presence of rehyped food, around 20 seconds after we present this food, mouse make the first eating bout. And few seconds later, we can see the increased calcium response while they're eating this high-fat diet. So when we look at the average calcium trace from the total number of cells, we can see both control ELT mouse showed increased calcium response in the presence of rehyped food at the first binge eating cycle. But it's hard to see the big difference before and after we add normal chow. However, at the last binge eating cycle, when ELT mouse showed hyper binge eating behavior than normal control, you can see only ELT mouse showed sensitively respond to that rehyped food exposure, suggesting that the sustained activation of leptin receptor lateral-epithelial neuron activity is maybe the critical for maintaining the hyper and sustained binge eating behavior of ELT mouse. So then we can think, what behavioral change would happen if we inhibit these neuron activities? So before we do that, we checked their efferent connection of leptin receptor lateral-epithelial neurons. So we found that these neurons project to medial preoptic areas and ventral tegmental area, and also the ventral lateral Ph.D. And these three target areas all are well involved in the modulation of food intake and weight gain control. And among them, I have particular interest in the ventral lateral Ph.D. because this is the primary source of endogenous opioid molecule, which is important for treating binge eating and obesity symptoms in humans. And also, very recent papers suggest that this brain area is critical for mediating some food intake and food-motivated behaviors. So firstly, we plan to inhibit the leptin receptor lateral-epithelial neurons projecting to ventral lateral Ph.D. And then we will see how we affect the obesity-prone feature of ELT mouse. So then how can we modulate the neuronal function in cell type specificity and also with position specificity? So firstly, we try to inject corticosteroid, which is potassium channels. So it can make a silence in the neuronal cell by making a hyperpolarization. But we express this corticosteroid virus with flip-dependent way, so it couldn't be expressed by itself into the brain of leptin receptor cree mouse. But we inject another virus, the retro AAV virus, expressing cree-dependent flip into the target area, the ventral lateral Ph.D. So it will allow expression of retrograde expression of flip onto the cell body of leptin receptor lateral-epithelial neurons. So finally, it makes it possible expression of a corticosteroid only in the subset of leptin receptor lateral-epithelial neurons projecting to the ventral lateral Ph.D. So after done this surgery, we measure their body weight when they exposed to high-fat diets. And surprisingly, we found that the heightened sensitivity weight gain of ELT mouse was totally disappear when they expressed the corticosteroid, meaning that the inhibition of leptin receptor lateral-epithelial neurons projecting to the ventral lateral Ph.D. can normalize the obesity-prone feature of ELT mouse. So this is the summary. We found that after exposure to early life trauma, they showed hyper binge eating and the high weight gain phenotype. And that is accompanied by downregulation of leptin signaling in the lateral hypothalamus. And we found that this is the critical factors inducing this behavioral phenotype. And also we found that the activation of leptin receptor lateral neuron activity is highly associated with hyperbinge eating behavior of ELT mouse. And also when we inhibit this neuronal activity, particularly the circuitry projecting to the ventrolateral pH, then we can block their obesity-prone feature of ELT mouse. So during the last few minutes, I wanted to introduce my future plan briefly. So I was curious to know whether we can expand our understanding to the different context of obesity and overeating issue over beyond the early life stress. So for example, there are many people who succeed at diet, like this guy. But as we know, maintaining weight loss is extremely challenging. So most of them go to the recurrent obesity with high vulnerability to weight gain and overeating issue. And that is called yo-yo effect because they get back to the previous state. So firstly, I thought that this sequence of yo-yo effect is very quite reminiscent of drug reinstatement in addiction model, like drug acquisitions, withdrawal, and drug relapse. And secondly, this behavioral feature, high vulnerability to weight gain and overeating issue, is very similar to the behavior phenotype of ELT mouse. So my question is that, what is the underlying neural mechanism of yo-yo effect? That is a very relatively overlooked before. So I wanted to challenge this yo-yo phenomenon by investigating the underlying neural mechanisms. So actually, I could replicate this yo-yo phenomenon onto the animal model system. The first group is normal control. They expose normal chow only. But the other group is exposed to the high-fat diet, so they're getting fatter. But the third group, these mouse exposed to high-fat food for 1 month, but later exposed to normal chow the other 1 month. So they get exposed to the weight gain and weight loss, which is the body weight cycling, to mimic the successful dieter. However, when we challenge them with high-fat food at this time point, we found that the mouse showing the yo-yo effect, they show the high vulnerability to weight gain compared to the other group's animals. And not only that, when we measure their food intake level, right after we expose the high-fat diet, during 2.5 hours, the mouse showing the yo-yo effect, they consume larger amount of food than the other animals during the short period of time. That is a compulsive overeating behavior or binge-like eating behaviors. And at that time, what happened in the brain? We found that the yo-yo mouse also showed activation of lateral hypothalamus in response to these compulsive overeating behaviors. So I realized that even though the ELT mouse and yo-yo mouse, they had different life history, but they had something in common in that they both have obesity-prone features and compulsive overeating behaviors. And I presume that the lateral hypothalamus may be important for mediating this behavioral phenotype. So in my future 5 years, I aim to explain the converged and shared neural pathway underlying these behavioral features. So I really thank to my lab members in UCSD. So Byung-guk is my mentor. He's such a wonderful mentor, and he supports and challenges me in many ways during my postdoc period. And Horia is postdoc. He contributed to do the electrophysiology work in the first part of a social project. Well, I'm a little bit sorry to him because I couldn't introduce all his nice data on today. And Chris is a rotation student, and during a very short time, he helped me a lot, and he contributed to do the leptin receptor shRNA study. And Baros, Shin helped produce tons of virus for this research. And also, Daniel is a former member, and he helped to characterize the lateral septum neurons in the previous my project. So thank you for your attention.
Thank you. We have a tight schedule because we have to get Sora to Blacksburg, but we'll take a couple minutes. But I just want to first make a comment. I think it was a very elegant presentation of a very sophisticated group of converging technologies, and I want to congratulate you on that. I was very impressed. Questions? Yes, Warren.
So I enjoyed your presentation. So I was wondering if you could, based on your work, particularly the obesity work, which is very fascinating, could you, is there a pathway to development of a translational intervention to help people with obesity based on your work that you could think of?
Yeah, that's why I want to focus on the ventral Ph.D. So especially the opioid system and opioid antagonists like naltrexone or naloxone is very promising treatment for the treating obese people or binge eating patients. But we don't know about the mechanisms, and also we don't know how the leptin receptor system is interact with opioid system. So if we find some neural mechanism and underlying the process, then it might be helpful to find the future therapeutic target.
What is the neurotransmitter of the neurons from the lateral hypothalamus to the periaqueductal gray? Are those glutamatergic excitatory neurons?
Actually, that is GABAergic neurons.
Okay.
But they also, maybe I can explain that tomorrow, but very interestingly, the leptin receptor lateral neuron also co-express neurotensin and other receptor molecule. And the neurotensin is a promising neurotransmitter, can stimulate the ventral Ph.D.
And the projection into the VTA, is that a distinctly different subset of neurons, or are there collaterals of axons from individual neurons going both to the VTA and into the PAG?
Yeah, that's a good question. Actually, you also check out, and that's not the complete separated. It's a little bit collateral. So, but in terms of amount of terminal, it's very different. But there are some small terminal we can see from the collateral projection. So in our future plan, we are going to stimulate or inhibit the terminal specific way to, yeah.
You'll tell us about that tomorrow.
Yeah.
Yes, sorry.
Really elegant work. So in either of your stories, do you know or can you speculate how these early life events downregulate either the Drd3 receptor or the leptin receptor?
Yeah, that's what I want to know. There are many studies how the environmental stress, especially the early life stress, can change the gene expression. And one of the possible mechanisms is the neuroendocrine system, because after they expose the early life stress, they show the hyperactivity of HPA axis, and because of that, it can change the some gene expression level in many brain areas. So
are those targets of GRMR?
Yeah, exactly, yeah.
Okay, I think we have to get Sora on to a next visit. Thank you very much again.
Yeah, thank you. Thank you very much.